All right, thanks everyone for being here today. So today, so my name is Wason. I'm an assistant professor in the School of Forest Resources at the University of Maine, and Laura roped me into working with Cedar a couple of years ago when I got started here at the University of Maine. I've been enjoying it ever since. And so today I'm going to be talking about some work that Laura and I have been doing together, looking at layering and what that kind of means for understanding the ecology of northern white cedar, but also what that means for understanding the management of northern white cedar too. So normally, when we think about reproduction and regenerating stands, we think about sexual reproduction. So the production of cones in this case that eventually release seeds out into the environment. You can see a picture of some seeds here that then germinate and grow into these very small seedlings of northern white cedar. This is a very delicate process in many places, so there's lots of opportunities for this process not to go well, including at these very small life stages for northern white cedar that you see at the bottom. And so they might be more sensitive to some big changes and swings in environmental conditions like we've been talking about relative to climate change and other effects. And so while this brings major benefits to the population because it brings in variation in genetic structure of that population, it does have some weaknesses. So many species have the ability to reproduce asexually. And so this is where layering comes in for northern white cedar. Layering is just vegetative reproduction from above ground shoots. And so in this schematic diagram, I'll be showing several that look like this. The green portion is kind of representing the above ground portion of the plant, the gray line representing the soil or substrate surface, and then the brown stuff kind of below there is the root structure. And when we think about layering, normally we imagine that a low branch from a tree comes in contact with the soil. And when it comes in contact with the soil, a species that can layer will grow what we call adventitious roots. And so that's what I'm representing with these thin brown lines in this case. When that happens, that's considered layering. And so we can see that when we pull out some trees from the forest, you can see they're actually often connected underground. And at first you may think that these are root systems that are connected, but I'll show you a little bit later how we can actually look at the anatomy associated with that. And we can better understand that many of these structures that are underground actually originated as above ground structures from stems. And that tells us that they're layers and gives us a little understanding of the past history of those individuals. So northern white cedar can do this really easily. I'm sure many of you are familiar with this concept, but northern white cedar can layer really easily, and we've known this for a while. So for example, James Curtis from the University of Maine, in his paper on preliminary observations of northern white cedar, noted that trees of seedling origin are probably the minority in the stands that he was looking at. Similarly, he did something similar to the experiment Laura mentioned that I'll be talking a little bit more today. If you take cedar branchlets and tear them off of the tree and essentially stick them in the ground, and come back a few months later, the majority of the ones that he did that on had up to as many as 16 roots per cutting in 2 months, and some of those roots were over 6 inches long. So that's just taking a portion of the above ground part of the plant and sticking it in the ground. And similarly, there's evidence from other parts of the region outside of Maine where these natural cuttings, so to speak, were being observed in forest understories. The horticultural industry has known this for a long time and takes advantage of this in order to produce large numbers of cedar for their industry. And we actually have an interesting new collaboration with a horticultural scientist, Brian Peterson at the University of Maine, that's helping us explore this a little bit more and to better understand what this means for forests ecosystems. But this begs the question, how common is this and how important is it actually in our stands relative to sexual reproduction? So these are some preliminary data from over 250 above ground stems that we pulled out of the ground across 4 sites from 3 different states. These are all lowland sites. And what we found, if we identify them in the laboratory, and I'll show you how we do that, but if we can confidently identify them in the laboratory, we found about 85% of the apparent regeneration in these stands were actually layers that were connected to each other. Only about 15% of the regeneration out in those stands was from seedling origin. So that suggests that understanding layers and how they relate to the ecology and potential management of these stands is really important. Similarly, if we look in at each of those individual layers, here on the x-axis, I'm showing the number of stems that were connected in a string. And then the y-axis is just the count, and then the different color of the bars corresponds to a different site. What we can see here, this is one individual layer, but you can see it's actually not attached to anything else, so that would fall into this one stem per string group. If there was another individual layer that was currently attached to this individual, then that would fall into that 2 stems per count, or 2 stems per string, excuse me. And so what we find is most of the layers out there actually look like seedlings. They're just one individual stem. Even if you pull them out of the ground, they apparently still look like a seedling to our eyes. But what we find, and when we look in the laboratory, is that they're actually all of that material below ground, other than the adventitious roots, is old stem material, suggesting it originated above ground at some point. And then the below ground connecting tissue, whether it connected out to a bunch of other layers, or whether that connected out to a branch at some point, that's often rotted away, and we can see evidence of that at the bottom. So how can we actually determine this origin and identity for layers versus seedlings? Well, the way that we've been doing is digging it up. And unfortunately, this leads to the mortality of all the individuals in our study. But you can dig it up and carefully excavate these individuals and try to collect them in such a way that if they are connected to other things, then you can bring those up with you. And the reason I actually started getting involved in this is based on using anatomy to understand these plants. And so what we know actually, so these are some fluorescence images that help us better identify different cell types. What we know about the anatomy of these species is they have this dark region of cells in the center here when imaged in this way, and those are the pith cells. Many of you are familiar with the concept of the pith that you find in the center of a stem, for example. However, roots, the below ground structures, do not have pith cells in the center. They have xylem cells, or the traditional wood cells, in the center of those. So therefore, we can look, and if we find evidence of these pith cells in structures that are below ground, that means that that originated as a stem at some point, and we can use that as a clue to understand if this was a layer origin, and sometimes we can even find the transition down to roots below ground. So we've been using this technique in the laboratory to identify these different individuals that we pulled out of the woods. Some interesting examples of the individuals we've removed from these stands. On the left-hand side here, all of these individuals are connected below ground. And so if you were out in the forest and you saw just the above ground structures, you'd say you had a nice little patch of regeneration there. When in reality, this is all one individual that's gone through multiple complex events where it layered in different ways and grew all these small, thin, adventitious roots, and it's continuing to layer. And that's part of why it's so important for us to understand layering and what that means for our management of these stands. So this is likely a sapling that bent over and formed many layers, but we also do find evidence of the more kind of traditional type of layering that we often think about. In this case, we have a long stem that apparently has a heel where we think it was probably connected to an above ground structure, maybe on a sapling or a nearby tree. And then this long stem eventually got covered with moss and was in contact with the soil, and it produced lots of adventitious roots. And now we have what appears to be several individuals regenerating from the end. But when you excavate it, you see that they're all connected back to the same stem underground. So just a couple examples of the complexity of layering that we do see when we're processing these samples. So that led us to put together a conceptual map that helps us better understand the different pathways that a seedling or a layer can go through different transitions before it recruits to a larger size class. So this is a relatively complicated diagram. I'll walk you through each of the 3 parts here, though. And we're primarily moving from the left-hand side here, a seedling, so something that comes from a seed and germinates. And then we're thinking about how can we move to the right-hand side of this diagram, which is recruitment to much larger size classes. So the first possibility is that it just grows fine and there's no layering involved. Another thing that we sometimes actually identified in the laboratory is that the moss layer thickens, and so that gray line essentially moves upwards, and you find these adventitious roots represented by the thin brown lines here that form, and technically that's a form of layering as well. However, what gets really interesting, and what we think we're seeing a lot of with our individuals, is there's something that causes this seedling at a very young stage to be laid down onto the forest floor. Maybe it's bent down by snow. We're still kind of working on figuring out some of those mechanisms. But when that happens, now that above ground stem tissue can actually grow some adventitious roots. And what we often find then is that the original root system, if it had one, is rotten away below ground. We very rarely captured this sequence where we were able to see the root structures underground. Remember, we can use the anatomy of those cells and those different structures to identify if they originated as a root or originated as a stem. And then what we think leads to some more of the complexity we see out there is that that happens again. So maybe it survives for several years in this first state and starts to grow tall again, but then something else causes it to bend down again. And now we actually can apparently have two layers that are connected, and we have two events of adventitious rooting here. And that can happen many, many times. And you all know how slow growing cedar is. You can imagine that it can be kind of stuck in this state potentially for 40 or 50 years if it's really struggling to get up to a larger size class, which it may eventually do. Then that brings us to the bottom portion of the graph, which is what we more often think about when we're first thinking about layering. So here now we're starting on the right-hand side with a larger individual. And we often think about, as I showed you before, that a low branch will bend down and come in contact with the soil, and that will then lead to the formation of a layer while it's still attached to the parent plant. We didn't often see that in our stands, and oftentimes the canopy of the mature trees was quite elevated, not such a way that it actually could produce layers by large branches in contact with the soil. What we're starting to explore now, though, is what if there's these broken branches that can actually fall from that canopy? How likely are they to land in such a way or in a substrate that allows a branch to fall from a canopy and root to the forest floor? There's evidence, as we know, of people sticking branches of northern white cedar in the soil and they can root, but how likely is it that a branch can actually fall from the canopy and layer? So that's something that we're kind of exploring now, and we're not really sure what the outcome might be. The reason a lot of these pathways are still kind of open questions is because the samples we have left in the lab usually don't have that original root system attached anymore. So even if we can say, yes, something is a layer, we can't say, well, it originally was attached to a large tree, or it fell out of the canopy, or it started as a seedling. It's very challenging for us to identify that last stage. So that's going to be the value of some longer term studies that might help us track some of these dynamics over time. So how should you think about layering when you're doing forest management? Well, one thing that some other studies have found is that layers can be more vigorous and acclimate to high light conditions and gaps, for example. So layers may be more resilient to some of the stresses involved in management. Part of the reason for that may be that layers have much larger root systems attached to them. So they have much better access maybe to water and nutrients and things that allow them to survive those stressful conditions, whereas seedlings, as I showed you before, have very small root systems and are a very delicate stage of that life cycle. This also suggests that when you're managing for northern white cedar, if 85% of your regeneration is going to come from layers, at least in our current state, you may want to focus on advancing releasing advanced regeneration rather than depending purely on regeneration from seed production. And so we're going to help explore some of that. We're actually doing a study now that Laura hinted at where we're just essentially jamming some branches in the ground, but we've actually got this set up in a pretty rigorous way where we're testing different sizes of branches from different individuals and different microtopographic positions and different methods of actually planting. And this is that exciting collaboration we have with Brian Peterson, who's a horticulturalist from the University of Maine, where he helped us take his perspectives on how he actually works with northern white cedar in the greenhouse and apply those out to the fields. We were really proud to get a horticulture scientist out into a cedar swamp. So stay tuned for the results from that study. We'll be wrapping up some of aspects of that this year, but also expanding and continuing it going forward. So with that, I just want to wrap up and thank all the field crew and all the students and others that have worked on these projects and helped to collect these data and make all this work possible, and of course the funders that this work wouldn't be possible without. And so with that, I'll pause here and I'll be happy to take any questions. Thank you.